Well, thank you, Larry. My job is to moderate the session, and I'm going to start off by reminding people that everybody is called Larry, and it's a little bit like the Monty Python sketch, if you can remember that far back, about the Australian university where everybody was called Bruce. And if you didn't know the name of the person, then you had to call him Bruce, because everyone was called Bruce. Well, here, Larry. So you meet a new person, you don't know what their name is, call them Larry, and you'll probably be okay. So that's the first thing. The other Larry, that Larry with the blue shirt, mentioned writings. Well, one of the things that one of our speakers, Tom, has done is written a book, and we're going to do a book signing of his book at lunchtime. I also happen over the COVID period to write a book about the history of biotechnology. So both of us are going to be signing our respective books at lunchtime after the session. And the proceeds, they're not many dollars each, the proceeds will go to the Gold Symposium. So you're supporting a good cause at the same time as buying a couple of books, which I hope will be an interesting read. I haven't read Tom's and he hasn't read mine, but I guess I will buy his and I hope he will buy mine so that we can actually do that. So the other thing I just wanted to say without further ado is in the talks yesterday, we were referred to as a higher organism, okay? And I kind of get uncomfortable with that because when I was looking after research at Sequana, we had an interest in C. elegans, and one of the guys that worked with us on C. elegans, I had made the mistake of saying, well, of course, we're working with this less complex organism called C. elegans, and he took me aside and he berated me and said, Tim, it is not a lower organism, it is a less complex organism. So I think when we're listening to these stories, we need to bear in mind that we are not the higher organism, we are a more complex organism in many ways. And I think, again, this morning you're going to hear more about how complex we are and how we're wired and how we're put together. And you're going to hear about that from Tom, talking about RNA and RNA catalysis and where that has led us. And we're going to hear from Colin about some artificial intelligence activities that his company has been doing over quite a few years now. And it's actually nice to be able to introduce people who I at least know their names and have seen them in the press, and all the people on the program I do happen to know in that way. So you're always one step removed from a Larry, and with no more comment, I'll hand the floor to Tom. Be my guest, Tom.
Thank you, Tim. The Larrys always throw a great party, and it's not just the talks, right? It's the interpersonal interactions, and sort of the two halves of this coin are both so thrilling. So when I asked Larry Gold if I could invite myself to talk here, I was thrilled when he said yes. Now, the topic today is, yes, it's about RNA, but the more general topic has to do with the walls that we scientists have built up between ourselves and the general public. Or maybe we didn't actively build them, maybe we just allowed them to rise up. But in any case, they have become a real problem. The public confidence in science in this country is at I don't know if it's an all-time low, but it's certainly at a very low point. This does not help us in terms of public health, in terms of thinking about the complex technological issues that are bang, bang, bang coming our way all the time, that we hope that our legislators will find a way to deal with in a responsible way. And so, what can we do about that? And so during the pandemic, I thought about this, and first question is, why is it so hard for scientists to explain their science to the general public? And I think the answer is clear. It's jargon, which is actually a wonderful thing in the laboratory, right? I can go into my lab and say, maybe you should CRISPR that allele into the endogenous locus in hex 293 T cells. And the student knows what that means, and it would take 3 days for me to explain it to Jim Linfield, who's sitting here. And Jim's a really bright guy. So So the jargon is not intrinsically a bad thing, but it has its place within the domain, and that's also true within the investment management people, and in the automobile shop, they have their jargon too. But we have to learn to switch it off when we try to reach a different audience. And so this really came to light, in the vaccination situation during COVID. And clearly, our approach of telling people that they were a bad person if they didn't get vaccinated. Maybe that worked in some countries. You can see we don't rank very well, but it certainly did not work in this country. And so I thought, well, maybe if telling them what they need to do is not a successful approach, maybe we should just take a step back and explain to them a little bit more, share with them what we know and what we don't know about complex scientific topics, and perhaps give some of them a little bit more ammunition if someone says that the messenger RNA is an evil drug, that they would be a little bit better informed, and they might be able to say, well, I can understand why you might think that, but did you know, this, that, or the other thing. So the 21st century, I think, is the century of RNA. If you think about it, the first half of the last century was the era of physics. Then DNA was king between 1953, when Watson and Crick announced the double helical structure with the help of Rosalind Franklin, and then about 2000, the human genome was done. So that half a century was the era of DNA. DNA's old stuff now. DNA is, everybody's comfortable with it. It's still useful, but the really exciting molecule is ribonucleic acid, right? So I thought, why not try to share the wonders of RNA with the general public? And why are people so comfortable with DNA? Well, first of all, they know that they inherit some of their personal traits through their DNA. They know that if they have a genetic disease running in their family, that is something that is within their DNA. They know they can trace their family history. If they're into genealogy, they know that forensic scientists can solve crimes with DNA, and they even use terms like, it's in my DNA. So why then is it so hard for them to go from deoxyribonucleic acid to ribonucleic acid? They must be sort of similar, right? In fact, they are similar. RNA is a single-stranded version of the nucleic acid, and just as the DNA, I'm going to try this finger thing here now, just as the DNA is held together by these pairing of the A's with the T's and the G's with the C's, those are the 4 letters in the DNA alphabet, the RNA, which has the same coding capacity as the DNA. It has A's, G's, C's, and U's, U being informationally equivalent to the T in DNA. Since it's a single strand, it can fold up within itself and make a huge variety of structures. Did that work? Oh, I should have used a different color. Larry, I'll practice before you invite me, before I invite myself back the next time, huh? Okay, so what gives me a sort of a foot in the door for this conversation is that by now most people have at least heard about mRNA. And remarkably, the mRNA in the COVID-19 vaccines, shown on the right in these greasy balls called lipid nanoparticles, is that these particles are really about the same size as the SARS-CoV-2 virus, which they are vaccinating us against, shown in the cross-section on the left. And they both contain RNA in an envelope. So if you know something about RNA, you know something about this battle between these two RNA units. Now, I think that part of the problem in getting people comfortable with mRNA as a vaccine is that a lot of social media, and also to those few people who still read magazines or newspapers, there was a lot of talk about mRNA as a medicine. mRNA is a drug. And that was frightening to a lot of people. And especially it's like it's a new drug, right? And, you know, it really should have been said that mRNA is natural. It's in every cell in our body. It's in all the food that we eat, whether you're a vegetarian or a meat lover, you're eating RNA all the time. In fact, you know, in Boulder, we might even say it's organic. So we've also known for 60 years how it works, right? This is not a newfangled thing. We've known that the information encoded in the double helix of DNA is transferred verbatim, that's a little bit of an exaggeration, some of you know, but pretty much into RNA. The A's, G's, C's, and T's in the DNA become A, G, C's, and U's in this single strand of RNA. And then groups of 3 of these building blocks along the RNA specify individual amino acids in proteins, and proteins are the molecules that are the movers and shakers in our bodies. They make our muscles move, they make our heart beat, they digest the croissant that you just had out in the hall that's in your stomach, right? And you notice what I'm doing here. I'm trying to pretend that you are all a general audience, right? And I'm trying to give this talk without using any jargon. And you can see that I am working pretty hard to achieve that, and that I am somewhat successful, but will get better, right, as I do more of this, right? And this is what I'm trying to get our students, our graduate students at the university, and my colleagues to be interested in doing as well. So, one way that one could make a particular protein to use as a vaccine, and a vaccine has to have a component of the virus that's like on the outside of the virus, so that it gives our immune system a heads up. So be on the lookout for this. If you see this again, it's a bad thing, you've got to attack it, right? So one way that you could make that particular protein, which for SARS-CoV-2 is the spike protein, would be to start with DNA and make messenger RNA and make the protein. But what the clever folks at BioNTech in Mainz, Germany, and at Moderna in Cambridge, Massachusetts, decided to do was just to use the mRNA, take a shortcut, and introduce the mRNA into the arms of people and let our bodies be the ones that would use the code in that mRNA to make the spike protein. And you know, one of the bad raps of the vaccine is that, you know, it wasn't perfect, right? Some people thought, you know, it really underperformed. Well, it protected against hospitalization and death. That's a pretty good thing, but it did not prevent against reinfection, which was annoying. And this is really not a property of the vaccine, a shortcoming. It's really a property of respiratory viruses. And they unlike the measles virus, for example, a vaccine just doesn't last for your whole lifetime. Also, the virus is sloppy when it's making copies of itself. And you might think that would be a bad thing for the virus, and many of those mistakes that it makes, in fact, are bad for the virus, but it then just lucks into some variants which turn out to evade the defenses that we've put up against them, such as the vaccine. And so there's this immune escape. But the great thing about the mRNA vaccines is they're so quick to go from concept to the vaccine that in principle, and we saw this a bit right with Omicron, one can just change the vaccine to match the mutated virus. And then also the fact that it has to be stored in the cold, in the really cold, is an impediment to distributing it to a lot of less wealthy countries where we would really like to be able to vaccinate the populations. And so that becomes tedious and expensive. But there are also some major advantages of these mRNA vaccines. I already talked about the adaptability. Do you know that the annual flu vaccine that we all take is produced by hand injecting chicken eggs with a destabilized virus to make it non-infectious, and that in the US alone, we use about 1 million chicken eggs per year to make just the flu virus. And this is such a slow process that you have to guess what kind of virus, flu virus, is going to be rampant during flu season and start injecting eggs before you know whether you're right or not. And as you know, some years they get it sort of mediocrely right, like 60%. Other years it's like 30 or 40%. Now with these mRNA vaccines, it's so quick that you could, in principle, wait until you know which flu is going to be rampant and then start making the vaccine. So that is something for the future, and also that's bullet number 1. It's not a 4, it's actually a bullet point. I don't know how to do this, but for me it worked, Larry. So so the first number 4 is that better flu vaccine. Second number 4 is personalized cancer vaccines are actually doing quite well in clinical trials. And this is perhaps something exciting for the not too distant future. How about therapeutic mRNAs? Because a lot of genetic diseases are caused by a mutation that either prevents a protein from being made, or on the other hand, makes a disabled protein or not very effective protein, right? Sickle cell anemia is a good example where the globin protein has this obnoxious property of changing shape in the red blood cells and causing incredible pain. So would it be possible to use therapeutic mRNAs to replace bad proteins with good ones? That's a high hurdle, because in some cases you need to make a whole lot of the protein, and vaccines, you need just a small amount to train the immune system. But the people are working on this, and it's exciting enough that there may be some opportunities for therapeutic mRNAs as well. So, am I done? Have I shown you that the 21st century is the century of RNA? No, I'm not done. Because this ability to encode proteins is old news, right? It's a new the mRNA vaccines are new applications of pretty old ideas dating from the 1960s, but it turns out that there are a whole bunch of RNAs that don't even care about the genetic code. They don't care which 3 units along the RNA chain specify which amino acids, because they are non-coding RNAs, and they have functions within the body which are thrilling and which I would like to share with you, because you are surrogates for my general audience. And because there are so many of these stories that could be told, I have just picked 3, and I'm not going to have time for all 3, so I'll do 2.5 of them. One is that RNA can fold up into a shape that it can do what we normally think of proteins as doing. It can be a biocatalyst. Secondly, RNA can bestow immortality. Wow, that's pretty cool, right? Well, at the cellular level, okay? Maybe not at the Larry Gold level. And finally, RNA can guide the editing of the DNA. This is the ultimate lording it over your parents, right? You can say, I'm going to go back and change you, your parent, right? So that's the CRISPR genome editing. So let's talk first about the catalytic RNAs. And this started in pond scum. When I came to Boulder, I decided to work on a smaller organism. I'm not sure it's even less complex than we are, but it's definitely smaller. But it's called tetrahymena, and it shares with humans the fact that its DNA is stored in the cell nucleus and it makes its proteins outside of the nucleus in the cytoplasm of the cell. And therefore, it's a reasonable experimental organism to understand basic features of biology. And why, though, choose this particular creature? And that's because it had 10,000 copies of a particular gene, a gene that makes an RNA that is part of the protein synthetic machinery, and that's a big deal in a cell, so you need a lot of it. And this cell divides very rapidly, so it needs to keep making these protein synthesizing factories. So 10,000 copies of the same gene. Now, most of you have how many copies of each gene? You know this. 2. One from mom and one from dad, right? So 10,000 is a big deal. And if you're a biochemist, which is what I am, you want to have lots of stuff to work on. And so we started looking at this gene, and one of the first things we found was that its sensible functional coding region, so to speak, was interrupted by a stretch of non-coding DNA called an intron. You can think of this as a commercial message that is interrupting your favorite film, right? You'd like to fast forward through this guy. That's not what the cell does. The cell doesn't have a way of fast forwarding, but instead it copies that intron along with the flanking useful regions into an initial transcript of the gene, and then it has to cut, and it's a little different than scissors, but it has to cut out that intron and patch together the functional parts of the RNA. And this is called RNA splicing. And when we found this in our genetic system in tetrahymena, it had already been found about 2 years earlier by Phil Sharp at MIT and a group at Cold Spring Harbor Labs in New York. And so it wasn't so novel to find an example of this RNA splicing, but nobody knew how it worked. And since we had 10,000 genes pumping out RNA that had to go through this process, this was an opportunity to figure out mechanism. And what we knew would be happening, or so we thought, was that there would be protein enzymes that would be cutting and pasting the RNA together in a very precise manner. And so we set up a reaction to find these enzymes. And we took the unspliced RNA, and you can see that the wiggly part is representing that intron that needs to be spliced out. And Art Zog, who did these experiments, mixed the unspliced RNA in a solution containing an extract of the nuclei of tetrahymena because we knew that the reaction took place in the nucleus. So this would be the source of the protein enzymes that were doing the splicing, and then we could purify them, we thought. And when we did this experiment and analyzed the results by a technique called gel electrophoresis, you can see the lane 0 is the material before the reaction, the starting material that still has the intron in the RNA, and the plus lane is with the nuclear extract, and it worked the first time we tried it, and worked the second time and the third time. It is very unusual in science to be able to reproduce a reaction so quickly and so easily. But there was a problem, and that is in a so-called control experiment where we left out some of the ingredients of the reaction, and particularly left out the nuclear extract, which is the source of the putative protein enzymes, the reaction worked just as well. Well, this was very disturbing. It was very concerning that the reaction didn't require any protein, and so there had to be some kind of an enzyme here because we had learned from James Sumner in his Nobel Prize speech in 1946 that all enzymes are proteins. And here was a reaction being sped up about 1 trillion-fold over the uncatalyzed rate. It was highly specific. There had to be a catalyst. What was it? So we kept trying to follow up the hypothesis that there was some contaminating protein in our reactions. We would boil the mixtures to see if we could inactivate that protein. Splicing kept working. Added detergent, splicing kept working. Finally, we were at a Christmas party when my graduate student presented me with this as a Christmas present. So, was there a protein that we couldn't find, or maybe there just wasn't one, and maybe the RNA was sufficient by itself to catalyze the reaction? And that turned out to be the case. And there was a whole community of people who were excited about this. And these were the people not working on Campbell's primordial soup, but thinking about the origins of life on the planet, which came first, the informational molecule or the functional molecule, sort of the ultimate chicken and egg problem, because we know that in our bodies today, you need to have an informational molecule so one generation can give rise to the next, right? That's DNA, but DNA doesn't do anything by itself, just sits there. You need to have enzymes to copy it, to make extra copies of it. So how did things get started if you needed to have the informational molecule, the copying machine there at the same time? And now we think that since RNA can both be the molecule that is being copied, but it can also be the Xerox machine itself and do the active copying, that maybe at the beginning there was an RNA world, there was RNA serving both information and function. Proteins came along a bit later, and DNA was a rather recent invention. When the total DNA content of a cell got very large, a more stable storehouse of the genetic information had a huge evolutionary advantage. So this led to the Nobel Prize in Chemistry, and I haven't aged a bit since 1989, so I'm sure you'll be able to recognize me in these photos. So that's one story of what RNA can do without worrying about coding. Well, how about bestowing immortality? This is the story of telomerase, and I have to introduce this by telling you what a telomere is Greek, telos is and mere, as far as I can figure out, is like stuff. So this is the stuff at the end of all of our chromosomes. Each chromosome has a linear molecule of DNA, so it has two ends, and they are vulnerable and they need protection, much as the plastic tips on the ends of our shoelaces protect the shoelaces from unraveling. What are those plastic tips called? Yeah, everybody. All these crossword people all know aglet. Aglet, Larry. You remember that, so that you can be the guy who answers it the next time, okay? It turns out that in most of our body cells, there's no way of maintaining that end of the chromosome. And so the chromosome shrinks every time the cells divide, and it provides sort of a measuring stick for how many divisions a cell is able to undergo. And after a certain number, like 50 cell divisions, cells stop growing. It's called the Hayflick limit, after the guy who first proposed this. And this is the way we just don't keep getting bigger and bigger and bigger, right, after we're an adult. Therefore, what happens in our stem cells that do need to keep dividing in order to replenish those tissues that we do want to, that wear out, such as, for example, skin cells, or also a lot of blood cells, which only last for 30 days and have to be renewed. So they need a way then to avoid stopping dividing. And what they use is this enzyme telomerase, and it contains, you must be able to guess it by now, what do you think telomerase has in it? RNA, right? So it also has a protein component in it. The RNA was discovered by Liz Blackburn and Carol Greider, who also won the Nobel Prize for the discovery of the telomerase enzyme. The TERT protein, which is now commonly used to immortalize cells in biotechnology, was discovered in my lab by a postdoctoral fellow called Joachim Lingner, who now runs a laboratory in Switzerland. And so this telomerase is the partnership between this non-coding RNA and this catalytic protein called TERT. And by building out the ends of the chromosomes, it allows our cells to proliferate continuously. It confers immortality. And before you think of that as being just a good thing, and a lot of people do think of it as a good thing, because you can buy products to elongate your telomeres on Amazon.com, you should realize that the other kind of cell, which is immortal, is a tumor. And so cancer, this telomerase is so important for continued proliferation that every single cancer cell has stumbled onto some way to maintain its telomeres. And usually, 90% of the time, that's by reactivating the telomerase, which was quiescent in the adult cells. And how does that occur? And so for a long time, we knew that it occurred, but we didn't know how. And the guy who figured this out was Levi Garraway, a brilliant scientist who was already famous at Harvard Medical School before he found, with his student Franklin Wong, who's now at UCSF, how this was happening. And it turned out that the activation of the telomerase took just one change in the DNA that was in the regulatory region that drives the production of the mRNA for the telomerase reverse transcriptase. And this is such an amazing mutation, it still boggles my mind. So 80% of all melanoma tumors around the world have exactly that same base pair changed in the gene for the telomerase, and it arises independently 400,000 times per year in unlucky melanoma patients, right? So that's how powerful of a cancer driver it is. It also is arising de novo in glioblastoma, in bladder cancer, in liver cancer. Interestingly, it's absent in a number of other really common deadly cancers like breast and colon cancers. They still reactivate TERT, and we still have to figure out how, because we don't know how this telomerase reverse transcriptase is activated in those cancers. It's not this mutation. Now, that's another amazing thing that RNA can do, but it's not the end of it. RNA can do something else, and if I had more time, I would tell you this whole story, but because of the time and because I want to leave time for answering questions, I'll just quickly say that this CRISPR genome editing is another story that involves a non-coding RNA, this time arising from bacteria that have developed ways to protect themselves from viral infections. And this is called genome editing. It's sort of like text editing, but the text that CRISPR is able to edit is the entire human genome, which in a normal font size and a normal sort of page geometry of a book is about 1 million pages of text. And CRISPR can find a single sentence out of those 1 million pages and then change one mistake, a U, for example, to an A. Very powerful, because in the laboratory, that's the kind of experiment we want to do to understand the function of a gene in many cases. But it's also of great medical potential, because there are so many human diseases, about 3,000 that have been identified, that involve mistakes in the DNA, and if we could correct those, and a few of the common diseases are sickle cell disease, cystic fibrosis, muscular dystrophy, potentially could be corrected by CRISPR. So, as you all know, this led to the Nobel Prize just a couple of years ago. Jennifer Doudna and Emmanuel Charpentier. Jennifer's going to be back here giving the Gamow lecture this next year, which is great. And she's also a graduate of our laboratory, and so we were particularly excited that she was a postdoctoral fellow. Here she is at one of our lab reunions. So anyway, I promised you that I would not take you through all of the wonders of this, but I do want to say that the ethical concerns should not be shortchanged the way that I'm going to shortchange them right now. We could talk about them in the question period if you want, but there certainly are concerns, especially when a lot of books have titles like The Unthinkable Power to Control Evolution, The Awesome Prospect of Editing Humanity, and the chance to direct the future of the human race. And you might think that's hyperbole, but actually it is possible to do such things with CRISPR. And therefore, to not talk about the ethics is not only irresponsible, but will get us into trouble again as scientists if we don't find ways to talk to the non-scientific public about such issues. So with that, I will say that in summary, I have described how RNA is the molecule of the century because as a messenger, it can produce proteins that, for example, can be used as vaccines or other therapeutics, but as a non-coding RNA, it can catalyze reactions in living systems. It can confer immortality at the cellular level. It can change the code of the DNA itself, and this makes it really the age of RNA, and this is what I have tried to capture in a non-technical way in the book, which is going to, and I haven't seen it yet. I haven't seen the hard copy, so this will be the first time I see my own book, and apparently there are some here. So thank you for your attention.
Lots of time for questions. One there in behind the Larrys.
Jim.
Tom, great talk, and I completely agree with you. I just had two comments. First is, it's not just the general public that needs to have help from these talks. I spent a good deal of time yesterday looking up terms that were thrown up on the screen very quickly, and spent way too much time doing that rather than listening to the talk. So that's the first comment. We need to look at this when we're giving talks to all of our colleagues, not just the general public. And second, I don't know if it's just a problem with the way we present things. I think there's a lot of people out there that are working very hard against distorting science results.
Yes, well, I think we all, I'm not telling you anything new to say that social media has propagated a lot of ideas that never would have gotten traction in previous decades, right? But that's a difficult thing to ban or to really control. And so I think we have to live with that as the new reality and find ways to try to counteract some of those wacky views that you can always get reinforced on social media, because there's always somebody who will agree with any crazy thing one can come up with, right?
Tom, I have a question from the online audience.
Go for it. Yeah, I forgot to tell people that Larry's managing that.
Yeah, I've got, and I'm just going to read it exactly, because I think it's a communication
It's probably from
issue.
my wife who's listening in, so I'll try to give it a good answer.
Why didn't the zero control not have the splice form if the non-spliced molecule doesn't need protein or is self-splicing?
Yes, so that's a great question. So it turned out that there were some small molecules that are present in all cells, like just magnesium chloride, which are also required for the RNA to fold up. So you can, when you isolate the unspliced RNA, it is stable in that form until you add the other cellular sort of milieu, the environment of the cell, and then it undergoes the self-splicing. Great question.
Oh, let's see where we go. Lady there in the fourth row back.
Please.
Hi there. My name is Sarah Gray, and I'm an author and a patient advocate. And one thing first of all, thank you so much for your passion, and the way you're explaining this is so helpful. The fact that you're saying, like, we need to communicate better, like science to public. It's like the way you communicate, the medium is the message. Like you're already doing it. I appreciate that. One of the things you said at the beginning was we can't just tell people not to get vaccines. Like you should do this or otherwise you're going to get in trouble, like a moral high ground or whatever. So at the end of your talk, you talk about we shouldn't use CRISPR because we're going to have ethical concerns. Like, I was just wondering if there's another
I actually wouldn't have said that quite that, but yes, go ahead. We have to consider the implications of various kinds of CRISPR therapies, and some of them have already been approved, like for sickle cell disease. Exactly. And I think there the ethics is very much on the side of utilizing CRISPR to cure instead of just, so right now, sickle cell, there's no cure for it. You can only give people palliative treatments like aspirin or, but now we can actually cure it. So anyway, so it's a complex issue, but please continue.
Well, I was just kind of wondering to talk about the vaccine issue where it's not really helpful to say you should do something. It's like we have to help people want to do something, right? To motivate them to want to do it. And it just made me think of when it comes to CRISPR, yes, there's certain situations like sickle cell where it's like, oh, that's a no-brainer, we're going to use it. I wonder, like, what's a good way, and this is maybe rhetorical, but maybe not, like, what is a good way to help the public understand what does it mean to say we shouldn't?
Good, thank you. So I don't have the complete answer to that, but my strategy, and I also, this is true if you're going to have a difficult conversation with another person, right? The first thing you do is you listen before you start talking. You let them express what their feelings are, what their concerns are, because you may, if you just start talking, telling them what to do, you may be not even addressing what is really their concern. So the first thing is to listen. The next thing is to, express some empathy for their thought, even if you really think that it's not very wise, and then say, well, I can, understand how you might think that way, but did you know? So for example, with vaccines, a lot of people don't know that that prior to their approval, they are vastly more heavily scrutinized by the FDA and other regulatory agents than any other drug that is prescribed. The safety bar is astronomically high for vaccines. Why? Because they're being given to large numbers of healthy people and they're being given to children. And so there's vast data on the CDC website that tells you about exactly how many adverse effects there are with various vaccines, and the numbers of incidences that they have tracked is in the millions, right? So they can tell you out of 10 million people who've had the flu vaccine, how many people have had an adverse event. And then you can tell your friend, you can say, well, did you know that although there are some adverse events with vaccines, that the most common thing is that the adverse event is the same one that the people would have gotten if they had gotten the disease that they're being vaccinated against. So that most people don't realize that if they hadn't gotten the vaccine and had gotten the disease, they would have suffered from that same adverse event. So these sorts of information often can, it might not convince some people. People tell, when I give this talk, people tell me, they say, there's all these people who will never listen to you. I say, well, I appreciate that, but there may be some people in the middle ground who are sort of worried but, open to conversation. Maybe they will be swayed, and maybe they will in turn sway someone else.
One more. We've probably got time for two more.
Great, thank you. That was a tour de force of jargon-free presentation. Congratulations, that was really clear. I think you mentioned reverse transcriptase at one point, but other than that, it was completely free.
I'm still working on it.
I'm an organic chemist and often get, molecule envy when we look at RNA because of the beauty of some of the things you were talking about. Two of the extra things that are wonderful about it. One, which is an extension of the catalyst thing, is that there's the autocatalysis possibility. It can make itself again by templating itself, which is just fantastic.
Yes.
And the second is the beautiful shapes that it makes. And I was wondering, in terms of reach out, have the beauty of the shapes ever been used as art to reach people?
Well, yes, they have, and they're beautiful to us as scientists. I don't know whether they're beautiful to the general public, but we do have models of these fantastic folded RNAs, and people seem to enjoy looking at them. So that's a good point. We might be able to use that more than we are using it as an entry point.
Okay, last one.
Boyja.
Thank you, Tom. I understand now much better why you get so annoyed when you see slides that are cluttered with a lot of things, including jargon. So jargon is, of course, something to be avoided, but there's another thing that is important, which is presenting ideas in an orderly manner that begins at the beginning, has a middle, and then an end. So kids, before they're a certain age, cannot understand that we don't already know everything they do. So they will just, begin at some place near the end and jump back to the beginning and so on. And some grown-ups really never get over this. And that trait is highly overrepresented among scientists. And so it's not just the jargon, it's just the ability to have enough empathy to anticipate what the person you're talking to knows and what they don't know, so that you can walk them up to a point where they can actually get what you're trying to explain. So that's a piece that all of us, really need to think about.
Thank you.
Great. Well, thank you, Tom. Thanks for the questions.